Hi, I'm Jennifer Katcher and I teach biology at Pima Community College, and I'm here today to talk with you about gel electrophoresis. So gel electrophoresis is a process to separate molecules based on size, shape, and charge. And we're interested in using gel electrophoresis today to determine if we were successful with our PCR amplification of DNA. So we created a fragment of DNA that was 680 base pairs long, and at least we hope we did. So today we're going to use some of that DNA that was amplified, we're going to run it on a gel, and from that we'll be able to see if we have a fragment that is of the size that we expect. So the gel that we're going to use in gel electrophoresis, it looks like this and it feels like firm Jell-O. So it's a little bit squishy, but you can see that it holds together. And at the top are holes or wells where we're actually going to load our sample. What I want to show you first, though, is actually how to load the gel. It's a little bit tricky, and it's something that requires a bit of practice before you actually load your real sample because you don't want to lose that sample if you don't load it correctly. So the first thing I'm going to do is I'm going to use a micropipetter to take 15 microliters of practice dye. So in my practice dye, it's just dye plus a little bit of glycerin, which will help make the sample heavy so that it will sink into the well. So I'm going to, 15 microliters is kind of a lot. You may have smaller gels that don't need as much DNA, or you may be watching this virtually. A lot of people have different techniques. I like to call mine the pool cue technique because I kind of brace my finger over the tip here, and then I typically get right on top of it and I place the tip in the water. Now a little bit of the blue dye may come out when I do that, and that's okay, but then I very slowly move my tip to the top of the well, and then I very slowly start to release the dye. And the goal is to have the dye go into the well. It's a skill, it requires practice, and that's why we have practice gels. So I'll let you take a look at this, and you can see that the sample is almost exclusively in the well. Now we're ready to prepare our samples to load into the gel for gel electrophoresis. So I'm going to start with the sample that we have from our PCR reaction in the last step. So there's 30 microliters of sample in here, and I'm going to take 10 microliters of that sample and add it to this tube where I'm going to prepare my sample for gel electrophoresis. So here's 10 microliters added for that, and then I'm going to take 5 microliters of loading buffer. So this will just make it easier for me to see the sample when I load it. It has dye and glycerin in it. So I'm going to add that to my sample, and I'm going to prepare a negative control and a positive control sample in a similar fashion, and that's going to allow me to determine if the gel electrophoresis process is working correctly. So our positive control is one that has, we know it has DNA that is of the right size, that 680 base pairs, and our negative control is just water. So we know that if we see that showing up with bands, that there's some problem with our run. The next thing I want to do is go ahead and prepare the electrophoresis chamber. So I'm going to take our gel and place it in the chamber, and this gel is being stored in some TAE buffer, and we're actually going to fill the entire electrophoresis chamber with TAE buffer. So we'll just use what's in the baggie here, and then I have this big bottle of TAE buffer. And you always want to have the gel submerged when you load the wells, because that way the sample won't stick to the gel. Okay, now that our samples are mixed, we are ready to load the gel. Before I actually put the samples in the gel, I want to talk about the apparatus a little bit. We can see the gel in the electrophoresis chamber. We can see the electrophoresis chamber has a negative end and a positive end, and I've placed our gel with the wells facing towards the negative end. Now the DNA is negatively charged. Remember those phosphate groups in the DNA backbone, they're negatively charged. So the DNA is going to run towards the positive end of the electrophoresis chamber. So when the DNA is moving through the gel acts like a sieve. And so it's composed of multiple small pores. And the small fragments of DNA can move quickly through those pores, but the larger fragments move more slowly. So at the end of our run time, we'll see the larger fragments are closer to the well and the smaller fragments are closer to the bottom of the gel. The first thing that we're going to load in our gel is called a molecular ruler, and it is composed of fragments of DNA of different sizes that are known. So there's a 100 base pair fragment and a 200 base pair fragment and so forth. And we will be looking at our gel, we will be looking for our PCR products to show up somewhere between the 600 and 700 base pair fragments in our molecular ruler. So I'm going to go ahead and place my tip at the top of the well and slowly release the ruler into this first well. And then I will load my positive control into the second well. So after I loaded the molecular ruler, I went ahead and loaded the positive and negative controls. And now I'm going to load my actual DNA sample in this fourth well. Okay, and now we are ready to plug in the electrophoresis chamber. So I'm going to match the positive ends and negative ends and then secure it. And then I'm going to plug the electrodes into the power supply. And I have set the power supply to run at 130 volts for 30 minutes. And that should give my samples enough time to separate the small fragments and large fragments, but not so long that the small fragments will run off the edge of the gel. So I'm going to go ahead and press run on the power supply. And if I'm successful, what I'm looking for are small bubbles to come from the electrodes, and indeed I see that the bubbles are forming, so that tells us that our gel is running. So the gel has run for 30 minutes, and what I can see is where we loaded the molecular ruler. I can see two distinct spots where the loading dye separated, and I don't see that as clearly for the other samples. But what we want to see specifically is the DNA and how far those fragments of DNA ran. So we're going to have to use the transilluminator with UV light in order to visualize the DNA. So the gel contains a stain that will attach to DNA, and we can't see it now, but we will see it under UV light. Now, unfortunately, I can't show you that well in our camera, but what I can show you is a photo of what the gel looks like. So what you can see is a whole bunch of small fragments and large fragments on the left-hand side with the molecular ruler. And then you can see a dark band in our positive control. So that's showing our what it looks like when you have amplified DNA for that barcoding region. And then fortunately our negative control is negative. And then our sample we see matches up with the positive control. So that tells us that we were successful, that we successfully amplified the barcoding region of the DNA from our insect, and now we're ready to send it off to be sequenced. So now we're done with the wet lab portion of our DNA barcoding project. The first thing we did was we extracted DNA from our insect, and then we used PCR to amplify that DNA. And last we used gel electrophoresis to verify that our PCR reaction worked. And it did. That was the good news. So now we can send that DNA to be sequenced. And once we receive those sequences back, our next step is to analyze those sequences using DNA sequence databases and other bioinformatics tools. So stay tuned for other videos that will provide details for performing those analyses. Thanks, and I've enjoyed working with you today.